Let's get started. Catherine Bair. And we're going to hear about some good lambda biology. You have someplace you can hook that? I'll ring the bell at 2 minutes.
Good morning. I'd like to thank the committee first for allowing me to speak today, and I hope that many of you will find it very interesting that our lab is working on using lambda as a tool for studying protein-protein association. And before I get into the actual work, I wanted to review just a little bit one very important point about lambda infection, and that's that lambda can either be lytic or lysogenic, as Ping had spoken about before. And this depends on two major factors. One, host cell physiology, and more importantly, the multiplicity of infections, or the MOI, and this is the number of bacteriophage per cell. In this case, if there is one or fewer phage per bacterial cell, your chances are likely that there's going to be a lysis. It's going to go through a lytic life cycle. If there are two or more phage infecting the bacterial cells, or two or more phage genomes in the cell, your chances are that you're going to form a lysogen. And we're using this property to help form the basis of our tool. And in general, the one way you can go from lambda being in a lytic life cycle to being a lysogenic life cycle is to simply add more phage. The more phage you add, and as you get over an MOI of 1, you get to the point where you're going to have lysogenic cells, lysogenic life cycle chosen. In our system, we're actually sort of artificially creating a very small microenvironment where the MOI is greater than 1 by giving lambda a reason to associate with another lambda particle. And we're doing this by display. Many people have heard of phage display. They're using phage display for protein binding studies. We're using it in a slightly different sense. We are using lambda as a scaffold for displaying a particular protein, and then we're studying the interaction of proteins through antibiotic selection. Because if there is only one phage per cell, you're going to have a lytic response. You're not going to form lysogens. However, if you give lambda a reason to associate with another lambda at a very low MOI, our thinking is that you're going to get co-localization of two phage to a cell, and you're going to form lysogens. And we're using either kanamycin or chloramphenicol resistance to select for these lysogens upon protein association. And our first question is, well, A, what protein do we use for this display studies? And secondly, how do we get that lambda that display phage formed? And we're using here the major capsid protein D. And this protein is very attractive for our studies because it exists at 450 copies on the outside of the phage capsid. This allows for a highly multivalent expression of your display protein. This is especially important if you have proteins that may not bind very tightly. When you increase the number of proteins existing outside of the capsid, you have a greater chance of getting a stable, strong protein association and a result. Secondly, the D protein has unstructured N-terminal and C-terminal ends. So you can fuse your protein to either end. This is obviously very important if the N-terminal end of your protein is very important for binding, and you have to block that up to form a fusion. And lastly, the D protein, unlike many phage, the outside protein is added after scaffold formation. So there's no problem like with T7 where you have to have the fusion protein pass through a membrane. So you can have highly hydrophobic or transmembrane or highly charged proteins using the system with absolutely no problem. Second step, how do we get it into lambda? We start out with standard cloning, where you clone your protein of interest to the D protein in a plasmid, and it is then recombined into wild-type lambda carrying either chloramphenicol or kanamycin resistance using Cray recombinase. And we're using a host cell that has P1 Cray recombinase in its chromosome, and we're using lock sites, mutant and wild type, between the plasmid and then the lambda genome. And then we form these recombined phage that carry amp resistance from the plasmid. They also lose or lack the alpha complementation by picking up the D fusion in this region, and they produce a PCR product of the proper size. So when we have plaque purified display phage, we have a very simple protein association assay. And this is nice because it's simple and it's flexible. And in the buffer that you're using for diluting your phage or for doing the association, you can add anything you want: inhibitors, small molecules, activators. And it's also done in vitro. This is very important. It's free of cells and cell components. So you're in 100% control of what's going on in this reaction. In its simplest phase, though, we just take phage from a stock that's 10 to the 10th, 10 to the 9th. We dilute them separately. Then you add those two phage together. You allow them to associate at room temperature for a few minutes. This is just sort of allow the proteins to associate and get the reaction moving. Then you add your cells. And since you are in control of the number of phage you add to the reaction, you can control the MOI at which these phage are added to the cells. We're using here 10 to the 8th cells. If you want to put an MOI of less than 1, so you're selecting against phage that don't associate because they would go lytic, and you want to find the phage that are going to associate to their display proteins, you can add the phage at 0.001, 0.001 MOI, whatever your choice is. We incubate at 15 minutes at 37 degrees, not room temperature. And this is just to allow for the infections to take place. And then we select them on antibiotic plates. And this is because we have in the display phage either kanamycin or chloramphenicol resistance. You can use either or both antibiotic for selection. And they're incubated at 32 degrees overnight just because these are temperature sensitive phage. And the first set of data I'll show you is work with just the vector phage. So prior to recombinating, and when the phage are still contain the LAX-Z alpha complementation gene, they're not ampicillin resistant, but they do contain either kanamycin or chloramphenicol. We wanted to see how these are able to infect cells. So we added phage from an MOI of many zeros up through an MOI of 3. And we get sort of a typical lambda infection pattern where the number of lysogens doesn't start to form at a significant level until we're at about an MOI of 0.1 or 1 and then above. And in this graph, we're selecting, for simplicity, just the kanamycin-resistant lysogens. And as you can see, the blue line is when only the vector carrying the kanamycin resistance gene is infecting the cells. And the red line is when we add both of these vectors together. And the slight shift to the right is simply because there are 2 phage per cell instead of a single phage. And as you can see, lytic in this region, lysogenic in this region. However, when we add a protein to the outside of the phage capsid, and here we're using acidic and basic aptamers as our display proteins. These are very simple little stretches of glutamic acid or arginine with glycine. And we're hoping that, well, through electric charge, they'll be able to associate, and this will help prove the principle of the system. And as you can see here, and again, selecting for one antibiotic only, the base phage, which is carries the kanamycin resistance gene, has a profile, low lysogeny frequency profile, very similar to the vectors. However, when you add its binding partner, the acidic peptide, you get a very large shift to the right. This represents a greater number of lysogens formed at a lower and lower MOIs. And this we're saying, oh, this looks like these proteins are associating. So at a very low number of input phage to the number of cells, we're getting lysogens because they are associated. They're co-localizing to a cell. You're forming a very microenvironment of a higher MOI. This is what we expected. However, these are simple little aptamers. We wanted to see, well, does this work with quote-unquote real proteins? So we got from Dr. Gali Prague the Q and ubiquitin proteins. Ubiquitin is a very small peptide, and Q in here is a very small domain of the VSP90 protein. And they are known to associate Q as a dimer with ubiquitin in about the micromolar range. It occurs through hydrophobic interactions. So we made display phage displaying these 2 proteins. And we got as a result very similar to the aptamers. Again here, the vector as your baseline, and the Q phage carries kanamycin resistance by itself, has a profile very similar to normal lambda, wherein you don't get a number of lysogens until the MOI is near an MOI of 1. You add its binding partner ubiquitin, and again, a large shift to the right, showing a very large increase in the number of lysogens you're getting at MOIs far below 1. Again, this looks like there's association. However, we can't say that for a fact this is a specific association. It could simply be there's 2 proteins being outside of lambda. They're associating through some other mechanism. So we did 2 very basic studies. The first is direct inhibition. We took the acidic and basic aptamers that are displayed in the phage, we made real full peptides, free peptides out of them, and we added them to an acidic and basic pair. And up here, we're just showing that it's an arbitrary number of lysogens in a particular MOI in our studies. And without increasing or decreasing the number of phage, we just added competitors of acidic and basic or a mutant version of the Q protein. And as you can see, the acidic and basic aptamers had a dramatic effect. They were blocking up the acidic and basic aptamers displayed on the phage, and they're basically inhibiting their ability to interact with each other. They're taking away the reason for lambda to associate. These lambda display phage suddenly become wild-type lambda in their lytic and lysogenic decision. However, the mutant Q protein, which is a large protein that's more related to the other system, its presence had no effect on the number of lysogens formed with this pair. That's one point of data showing that these are likely specific. Then we looked at the quote-unquote real proteins. In the ubiquitin Q pair, again, we are being inhibited by the ubiquitin and Q-free proteins. They're blocking up the sites available in the phage for them to interact with each other. However, the mutant Q, as I mentioned before, this is a mutant that has only one amino acid change. It loses all its ability to bind to Q itself or to ubiquitin, and it has really no effect on the binding partners. It adds a little bit of noise to the background. And likewise, the acidic aptamer, which has nothing to do with these proteins, that was able to inhibit the acid and base pair, does not have any effect on these proteins here. And the second set of studies we did, which is very indirect, we just wanted to see if we took a lambda library, sort of a random library, so we just made a bunch of display phage that display open reading frames from actually a human library. We wanted to see if this library would interfere with the number of lysogens we get from the pair. So we kept the ubiquitin as a single input. We increased the number of Q. We got a number of lysogens. When we added this library, we thought, well, if it's nonspecific and it's just random interactions, you're either going to get a greater number of lysogens, a whole lot through the roof, or you're going to completely inhibit the pair. And there's really no difference between the two of them. So we thought this is another point for specificity. And since this system seems to be very simple, it seems to be very specific, it's also done in vitro, away from cells, and it works with homodimers, multimeric complexes, small and large proteins, we're looking at and studying now using this as a library screening technique. A lot of people use biopainting now for library screening. This is a very quick and easy way to screen out binding partners. It can be used in a high throughput format. We're also looking at the use of mediators. Many proteins only interact in a 3-protein complex or with other activators. And we're also hoping to look at improving the affinity of small chain antibodies. If you can get more and more lysogens as the MOI goes down farther from 1, that could possibly relate to a higher binding affinity. And lastly, some work we've done with drug screening using very small molecules to inhibit pairs. This is very important in all sorts of AIDS studies or in cancer studies. You need to be able to have a high throughput system that's quick, cheap, and easy. And lastly, I'd just like to thank the people who have helped greatly in this project. First is Amos Oppenheim, many of you know him. Gali Prague was the researcher who supplied us with all the ubiquitin and Q reagents, and then my chief, Dr. Shankar, as well as my lab, and 2 of my lab members are here at the conference, Dale Lewis and Fuchsia Gal. They're presenting their work the next couple of days, so please stop by there. And I'll open up the floor to questions. Okay, you got a microphone, sorry.
So how tight does the interaction have to be? How long-lived?
Well, the interactions we worked with right now are Q and ubiquitin, which is the micromolar range, as well as some cancer proteins, S100 beta, p53, MDM2. Their association is a nanomolar range, and the effect there is very dramatic. The aptamers are completely uncharacterized. We're not sure what their interaction what their level of interaction is. But we haven't worked with any protein that's a fleeting interaction. We haven't seen that yet, but we assume that the number of lysogens you get will go down because if it's a fleeting event, there's just not enough time for them to be together to infect a cell. So once we start working with more and more proteins, we will be able to get a better idea of does the actual binding really shift the slope over to the right in the graph, or is there just sort of you can't quantitate it, it's either there or it's not.
So you mentioned, I think, that Q was a dimer?
Well, Gali showed that Q can bind to ubiquitin best as a dimer, but it can bind as a monomer with a lower affinity.
So the question is, can you see the Q interaction also if you have 2 forms of Q, one with CAN and one with chlorphenicol?
Yeah, the Q by itself, it has a slightly better binding affinity slightly better slope than if you were using just a plain vector phage. We have seen that. But that, I guess, would count as sort of a fleeting, very weak interaction because it's not stable. Mm-hmm. It's not, because there's a nice tight hydrophobic interaction between Q and ubiquitin, but to Q, it's a helix to helix. it's not that tight.
If I understood your inhibition assay correctly, when you had the Q-ubiquitin interaction and you added Q, you lost lysogeny.
Mhm.
But even in an MOI of 10, you didn't regain lysogeny. Do you know why? Wouldn't you expect it to come back up?
We
as you're
didn't
putting-
do we did the study where we had a stable input of phage. So there was, I think it was like 10 to the 5th phage in that actual assay. And we didn't change the number of phage, we just increased the competitor. But I have done the study before where we had greater numbers of lysogens we were looking at. So we had in the 500 range. It just simply took more competitor in the micromolar level. Hey, guys.
Have you found whether there's an upper limit to the size of the protein that you can fuse to D, at which point lambda starts losing functionality or anything like that?
Well, our limit right now is how much we can clone the genome, because we have about a 2.5 kb limit. And the largest protein I've gotten in there, and it's perfectly stable and fine, it seems to be, is the MDM2 protein, which clones at about a 2.2 kb gene size. We can also, since you can add D in trans, we could add the largest protein we could think of that wouldn't actually rip the head apart, because we don't have to clone it at that point. It's just a little bit dirtier of a trick.
Okay, I have a couple of related questions, partly related to that. There are, one can imagine if these two phages come together with random DD interactions, there are a lot of steric arrangements that would not lead to productive infections by either phage, or by at least one of the phages. So don't you also have to, won't you have to show, A, that the amount of D with each of these derivatives is going to be about the same on the phage, and we know they're distributed the same? And related to that, if you take two known associations that you can use, do these things actually scale in terms of their, you take your numbers you get from your lysogenization curves, and do they scale in some linear way? One would hard to believe they would.
It's well, there's
It would take me half an hour. So
there's a lot of questions in there. The first one is about you have phage coming together. Yes, their tails are going to be whipped in all sorts of directions, but we found that more often than not we get aggregation. So we lose most of our productive pairs. So you can imagine there's not just 2 and 2 phage coming together. There seems to be big gluts of phage just interacting because the proteins are interacting. And when you do have, tail up here, tail down here, if you've got about 6 together or even more, you tend to lean towards, well, eventually you're going to get 2 of those or more are going to infect the cell.
that means it shouldn't scale then, right?
Yes, it's hard to scale because you've got one is the steric hindrances. We've used homodimers as well as large heteromeric complexes, and they work. They just work at they're less efficient is what it comes down to, because you have a lot of steric hindrances, you have a lot of other factors they have to negotiate in there. And when we're looking at just pure number of phage and how many of these lysogens we get out, there's a big decrease. Part of that is aggregation. Another one is the tails don't match up. But yes, we've got problems with that. But one trend we see in all of these is that as the KD gets smaller, the binding affinity gets higher, that slope in the graph keeps shifting farther and farther to the right. And so the stronger that they interact, the more interactions we seem to see, the more lysogens we get at lower and lower MOIs. It's really hard to use a system for quantitative reasons. It's more of a qualitative, do they bind? And that's when we're sort of in a murky area looking at the improving the affinity of proteins, because we don't know what our sensitivity limit is. And it's not a very linear curve. It's just either it shifts really far to the right or it stays closer to the vector level. It's kind of at the point we're at right now.
Roger? Do you know how many genomes are getting injected in these clusters? I mean, do you see more than double lysogens in these?
We see more than double lysogens. Just using PCR, we can find that there's some cells with only 2. There's some cells that have 2 of the same phage in there, or 2 or 3 or 4 of either one of the chloramphenicol or kanamycin phage that are in, but there seems to be a very scattered distribution of it. It does seem to be a very random event. And again, that goes back to aggregation, goes back to steric hindrances, and how many proteins you need to get the complex to work, such as dimers on a single protein or dimer on dimer interactions.
Okay, one last question.
I would try to play the role of Max Delbrück.
Oh no.
He would say, I try to understand what you said, And do I understand it correctly that when you have two phages which adhere to each other because you have on the surface interacting protein, then it increases much the chance of double infection? Is that correct?
Yes.
But what happens if they some sort are upside down attached, so their tails are not properly arranged? Would they still both infect?
Well, yeah, if you've got a phage that's literally 180 degrees to each other, so one is touching the cell and facing, the other one is way up in the air, you're likely not going to get a lysogen out of that particular setup. But again, if you've got aggregates of phage, when the MOI is extremely low, you tend to get just one-on-one. When the MOI increases a little bit, when there's association, you tend to get a jumble of phage. And there's also, there's interaction between the proteins on the heads. So there's some play, like Q and ubiquitin, what I call the real proteins in these two pairs. They can move and they can flux. You can actually have movement as that's going along, and you can have there's obviously the attraction of the tail to the cell, which may make a difference in phage coming down towards the cell, simple Brownian motion movement in the solution, things like that.
So there is long enough, the linker, so they could rearrange themselves.
Yeah, the linkers are about 7 amino acids long, and they seem to be very flexible because we've gotten large proteins able to interact with each other even in large complexes. So that's why we're looking at the use of mediators, because we think if we have enough play between the phage head and the protein that's being displayed, that if a mediator is in there, it will have an effect. The two proteins will be able to swing enough to make their matches. It's not perfect, but it seems to be working pretty well.
Okay, that's very cool. I think we have to move along.